great to be here. And so today what I decided to talk about is kind of give an overview of one of the main areas of research that I've been working on in the last several years. And that is comparative phylogenetics and moving into phylogenomics, biogeography, and also how these two kinds of studies can lead into affecting taxonomy and conservation in neotropical primates. And so just to give a few definitions up there for people who might not be as familiar with these topics, phylogenetics is the study of evolutionary history and relationships among individuals and species, and usually the way that results are represented in phylogenetics are with trees or phylogenies. Phylogenomics is just adding in genomic data, so instead of phylogenetics can really be done with either a single genetic markers or a few genetic markers, or it can even be done with a morphological data set. You can construct a tree to understand relationships across individuals or other kinds of data sets. But with phylogenomics, you're starting to have sampling across the genome or even whole genome sequencing to look at phylogenetic patterns. And then biogeography is the study of the distribution of species and ecosystems in geographic space and through geologic time. And this biogeography can be modeled on the tree to look at how populations or species are moving across space and time. And then neotropical primates are my main area of study, and they're a really excellent study system because neotropical primates all stem from a single common ancestor. It's thought that they rafted over from an African population maybe around 30 to 34 million years ago. And so all the diversity, all the standing diversity that you see in the neotropics for primates all originates from one single common ancestor. And so there have been multiple radiations, some of which have gone extinct in the neotropics, and the common ancestor to all living neotropical primates is from about 20 to 22 million years ago. To give you a sense of that diversity, we can divide, this graphic divides it into 3 families, or we can also divide into 5 families, which is the squirrel monkeys and capuchin monkeys are the Cebidae, the night monkeys are the Aotidae, the marmosets and tamarins are the Callitrichidae, the spider monkeys and howler monkeys are Atellidae, and then at the bottom, TD monkeys, wakaris, and sakis make up the Pithecidae. Okay, so this kind of shows the standing diversity of the platyrrhines or neotropical primates. And another way to represent that diversity is just to look at the variation that you see in neotropical primates. So one of the things more broadly that I'm interested in is understanding the morphological and behavioral diversity of neotropical primates. But for this particular study, when I'm trying to, or this particular group of studies that I'm going to show you, I'm trying to understand what the evolutionary relationships are among all of these different taxa, and also how they've spread across the landscape of Central and South America over time. You may have been at a Beck talk a couple of years ago where my postdoc, Charlene Santana, who's now at the University of Washington, she talked about facial pattern complexity evolution in neotropical primates. And so this is a similar idea of using a phylogeny and marking on character states to try to understand how things have changed over time. And that's what I'm going to be doing here today also, but using geography, using change in regions of occupation over time. Okay, so that means we need to think a little bit about Central and South America. And so if we look at South America, if we focus on South America, some of the major attributes of that continent are the Amazon basin and the Atlantic coastal forest. These are the two tropical and semi-tropical rainforest habitats, and what we may think of immediately when we think about primates. We may expect to see them in these tropical rainforest habitats. But in fact, you can see primates also up in the Andes on the west coast of South America, and also in these drier habitats like the Llanos, Caatinga, Cerrado, and central grasslands. And so part of what I'm asking today is, how have neotropical primates colonized the Amazon, these savannah-like habitats, and the Atlantic forest? And just to also give you a sense of the diversity of neotropical primates, they range in size from the tiny pygmy marmoset that you could fit into your hand to the ape-like muriqui. And their diets also are highly diverse. So you have fruit, insect, and omnivores, as well as seed predators, gumivores, and leaf eaters. But it turns out with human activity and anthropogenic change that if you start to look across neotropical primates, about 45% of standing taxa at this time are considered threatened. So they're red listed, and that means they're either considered vulnerable, endangered, or critically endangered. And I put up an extreme example here of the spider monkeys, and what you can see is every single one of them listed here are on the red list. They're all threatened with encroaching, with extinction near in the future. Okay? And also what you can see, and this is especially true here, I'm looking at Amazonian marmosets, Miko. What you can see is there's still a lot of work that needs to be done. If you look at these taxa, you can see for their status, about half of them there are data deficient. We don't even know enough about these species and their distribution and their population size to even say where they should fit in terms of their conservation status. My own research spans across the Neotropics, but my primary focus has been in Brazil. And this is just a kind of a pop-out map of Brazil showing where the threatened species are concentrated. So every pin put in there, if it's red, it's critically endangered. If it's orange, it's endangered. And if it's yellow, it's vulnerable. So they're all red-listed taxa. And what you can see is what might be expected if you think about it, that there's a concentration of threatened taxa in the Atlantic coastal forest. That's where Europeans first colonized Brazil, and that's where for hundreds of years they've been having high anthropogenic change. So it's a relatively old invasion, and when people started thinking about conservation and primate biology, it was already devastated. So there's just about 5% of the original Atlantic forest habitat left. But what you also see, which might be more surprising, is that you start to see across these dry areas and also in the Amazon that there are also threatened taxa and even critically endangered taxa. Okay, and so those changes are much more recent, and those are changes in terms of the anthropogenic changes that are accelerating in a lot of places right now. So this is kind of a imaginary idea of what the habitat might have looked like at some point in time. But when we start to dig down into what things look like now, we see that much of this habitat all across South America has actually been modified from these original ecosystems. Just to give you a sense of that, this is Atlantic coastal forest, and you can see the patchy quality of the forest because much of it has been cut down for farming and cattle ranching and so on. This is just a picture from some of the Cerrado habitat, the savannah-like habitat, and it's being affected by both human use and by drought conditions that have intensified with climate change. But then if you also if you look at the Amazon, which we may think of as the last pristine area that's still intact and huge and so on, when you really drill down there, this is in the east of the Amazon in the arc of deforestation, and you see basically a patchwork quilt. But even further, deeper in the Amazon, for example, here in Acre, what you can see is roads lead to this fishbone cutting, and so you end up with highly fragmented forest, even deep in the Amazon as well. So I kind of put that preface just to say that things are changing as we work on these questions, and the need for understanding the biodiversity within Neotropical primates just increases. But also the good news is that there's more and more focus with a small but growing group of researchers that's really interested in these topics and has recently come together to work on these questions. And so this is a picture of participants in a symposium I put together in 2012 that was the first symposium on biogeography of Neotropical primates. And as a direct result of that symposium, I was invited to be lead guest editor on a special issue of Phylogeny and Biogeography of Neotropical Primates in Molecular Phylogenetics and Evolution. And so this has kind of been a watershed moment for really getting a lot of papers out and getting a lot of interest in these taxa and their relationships. And so this is kind of a summary slide to show what work has been done up to now. And the great news, you can see that most of these papers are quite recently published, but the good news is we're covering almost all the taxa now, and looking at taxa that had never been considered before. Although there's a few missing still, like the muriqui, the wakaris, and the saki monkeys. There's also been some really important recent pan-platyrrhine studies looking across platyrrhines at phylogeny and biogeography, and I'll talk about a few of those as well. And so some of the biogeographic questions that I'm going to talk about today are things like, what explains areas of overlap or sympatry between seemingly closely related taxa? And when did modern primates colonize different major regions of the Neotropics? So you can use these tools to try to ask, these are just a small sample of the large number of questions you can try to answer with these data. And as I said, I'm going to give a broad talk. I'm going to show lots of different examples. And what I want to say here in terms of methods is that many of these different studies that I'll show you have similar methodology in common, which is kind of this general picture, that you want to collect samples from known locations from animals that are of known taxonomic identity. Okay? So many of the studies before this last 10 years have just taken samples from animals from zoos where they don't really know where they came from. Okay? And then the next step is DNA extraction, amplification, and sequencing. And then to create these phylogenies or trees, you can use maximum likelihood and Bayesian methods for phylogenetic reconstruction. You can also anchor the tree or calibrate the tree using, for example, fossils, and then estimate the divergence times on the tree. And then you can also use these new statistical methods, these models, for reconstructing ancestral geographic state. And I'll show you some examples of beast phylogeography and also biogeobards. And so basically what I'm going to do in the talk is talk longer about capuchin monkeys. That's the group that I've worked with the longest. And I kind of want to show you the move from starting with just single mitochondrial markers and working through to have genomic sampling and then whole genome sequencing. So you can see this progression over time in the field in terms of genetic to genomic sampling. But then I'm also giving you a quick view of some other radiations of Neotropical primates and what kind of picture we see about them from looking at the phylogeny and biogeography. And then I'll talk a little bit about comparative biogeography. How do you pull all this information together and have a broad picture of what's happening across time and space with Neotropical primates? And finally, I'll get to taxonomic and conservation implications. How does this matter? Does this actually change how we think about conserving and making priorities for among the different Neotropical primates? So I'll start out with this paper, which came out in 2012, and this was an international collaborative effort where we were getting samples both from museums and from the field. And what we were looking at in this paper was the radiation of capuchin monkeys with the hypothesis that there are two types of capuchin monkeys within the group capuchin monkeys. And so those two kinds of monkeys are here on the left, gracile capuchin monkeys, and this green here is the sympatry between the two types. The other type is the robust capuchin monkey. So you can see the robust capuchin monkey spans the Amazon and all the way to the Atlantic forest, whereas the gracile capuchins, if you look at all the different species, they're throughout the Amazon as well and up into Central America. So one question in this first paper was when and where did capuchin monkeys diversify, and what explains this sympatry across the Amazon of these two kinds of capuchin monkeys? And so just to give you this example of having fossil calibrations, so in this study we had 5 fossil calibrations to anchor the tree and assess the divergence time. So each of these red circles is a different node that's calibrated by fossil evidence. And from that, what we found was, in fact, the gracile and robust capuchin monkeys did form 2 separate clades, and that they diverged about 6 million years ago in the late Miocene. Okay. We also did this biogeographic analysis. This was in Beast Phylogeography. And here, what you can see, again, this is showing the 2 clades of sapajous, the robust capuchins, versus cebus, the gracile capuchins. What you can see is the warm colors, the reds, are Atlantic forest, and the blue and greens, the cool colors, are Amazon and Andes. And so what this is showing is through time, starting from 6 million years ago at their divergence, that the sapajous, the robust capuchins, are modeled to stay in the Atlantic forest until about 500,000 years ago, when they colonized the Amazon, where cebus has been diversifying for the last several million years. Okay. So our hypothesis from that paper is this idea of vicariance between cebus and sapajous, with cebus diversifying in allopatry in the Amazon and sapajous in the Atlantic forest, until there was a re-invasion of the Amazon by sapajous and a re-entry into sympatry with cebus. Okay? And so what I'm going to show you now is our follow-up studies to that original study, and this was led by Marcela Lima in Brazil and Janet Buckner, who's an EEB student here at UCLA. And what we did for this study was to triple the number of samples. And so here you can see our sampling for cebus at the top and some of the diversity within cebus next to them. And then on the bottom you see the map with the sampling for sapajous and some of the diversity here as well. So we wanted to especially look at the pattern that we could find for this rapid reinvasion of the Amazon. With more in-depth sampling, do we still find the same pattern of sapajous diversifying first in the Atlantic forest and then spreading rapidly into the Amazon basin? And just to give you a sense of, and this is going to become important in terms of the taxonomic implications. So right now, the IUCN recognizes 8 species of sapajous, 8 species of robust capuchin monkey. And you can see there are a couple in the Amazon, Macrocephalus and Apella, then Libidinosus and Kai are in the dry savannah-like habitats, and then Flavius, Genthosternus, Robustus, and Nigritas are all in the Atlantic coastal forests. Okay. So this is a Mr. Bayes phylogeny with all those samples that I showed you, and each triangle is a clade that represents multiple tips that are in that single clade. And so what we found here again was a clear division between cebus and sapajous. And then here what we have, I want to focus on this part of the tree. So all of these specimens here are Atlantic forest capuchins, and then we have this subclade sapajous clade 4. That's everything else. That's sapajous spread everywhere else. And so what we wanted to do was drill down and look at the diversity of this clade 4, this cosmopolitan clade. And what we found was, so each of these colored clades is represented here by that same color. What we found was, in a nutshell, we found geographic structuring for the sapajous clade 4 across this wide distribution, but it didn't correspond to the species named by morphology. And so you can see, for example, this clade here includes Macrocephalus and Apella. This also includes Macrocephalus and Apella. This includes Apella and Chi, and so does this. And this one includes Apella and Libidinosus. So what that means is there's geographic structure, but it doesn't match the morphology. The genetics don't match the morphology. We also did this beast phylogeography analysis with the extended sample set. And here we have the Atlantic forest as the outgroup. And then we're looking at that rapid radiation everywhere else. And here again, this is saying at 400,000 years ago, the capuchins were able to extend their range out from the Atlantic forest to Rondônia, which is the southeastern part of the Amazon. And then you can see rapidly moving across time to the present, this spread from Rondônia up through Guyana to Inambaria, from Inambaria through the Negro to Guyana, from Rondônia 3 different times, if you see this red, 3 different times spreading to savanna habitat, and 1 time spreading back to the Atlantic forest. So again, this is even stronger evidence for rapid, explosive radiation where, if we think about this in terms of biogeography and geographic barriers, suddenly we have this kind of monkey that's able to cross geographic barriers that stop other monkeys with ease now. It can move across the Amazon River, it can move across other tributaries, it can move and invade into dry habitats multiple times, and so on, okay? All in a short period of time. Okay. So one question was, is this pattern we see that doesn't match up against morphological data, is this just something funny that's going on with mitochondrial data? Because all this data that I showed you up until now was using mitochondrial markers. And remember that mitochondrial markers are inherited from the mother only. And what they're really showing is how females move over time and space. Okay? And so what we did was we did a parallel study using a genomic markers, ultra-conserved elements. And these are highly conserved regions of the genomes. And what you can do is you can target these UCEs, but then sequence the flanking regions that are more variable. And so in that way you can use the flanking regions to look at genetic variation across individuals and populations. So we did this genomic sampling with UCE data to analyze a subset of the same data set and see if we came up with similar or different results. And so what it turned out with the UCE data, and this is just here again, we have the Atlantic forest group as these encompassing clades right here, and then one subclade of the Atlantic forest group is everything else, this big widespread cosmopolitan clade. And then if you start drilling down to the subclades within that, again you see here Apella, Chi, and Macrocephalus all make a clade. Here Apella and Macrocephalus make a different clade. Here Flavius, Apella, and Libidinosus make a third clade. So again, you don't have the clades map onto geography, but they don't map onto the morphological hypotheses for what the species actually are. And so our interpretation of these data, and we found that the UC paralleled the mitochondrial DNA in showing this same pattern. What is that there's gene flow across these regions, and there is genetic drift and or response to environment. So you see morphological variation across this broad region, but it doesn't represent genetic isolation. These are not distinct species. They haven't had enough time, and they're too able to move back and forth across different areas to truly speciate and be different things. And so from this work, we suggest that sapajous should be considered to have fewer species than is currently described. And our suggestion would be something like this, where that entire Amazonian and savanna habitat would be considered a single species, Sapajous apella. Okay. And just to show you kind of the future, what we're doing now is whole genome sequencing. So there's currently just published a 80x Cebus capucinus imitator de novo genome available on GenBank, and our group, I along with Patricia Izar at the University of Sao Paulo, we've just finished 30x resequencing of 9 other capuchin species genomes, and so now we're just getting started on analyzing the differences across these different species. And just to show you the very first look at difference. So these are all SNP and indel recovery compared to the Cebus capucinus imitator genome. And so what each bar is showing is the percentage of variance across all map sites for each of these taxa. And what you can see is kind of the same pattern that you would expect if Cebus is a more recent radiation and all the Cebus individuals are closely related to each other, and then Sapajous as a whole is that radiation that's 6 million years separated from Cebus. So you can see Cebus is about 0.5% variation within that group, and then compared to Sapajous, Sapajous has about 1.5% variation compared to Cebus capucinus, and that's about what you see with humans versus chimpanzees. Okay, now I'm going to just quickly go through some other examples of studies that we are doing with other neotropical primate taxa. This one is on the marmosets and tamarins, led by Janet Buckner. And here is another question, a similar question as we asked with the capuchin monkeys. What explains this overlap in range between these small-bodied tamarins who are found in western Amazon and these large-bodied tamarins that are found up by the Andes and then also all across the Amazon. So this area right here is an area in Sympatry. And the thing that's interesting, again, going into this study is that these little tamarins, all of them have this saddleback. The smaller sized ones across this range don't have that much morphological variation, and so they've often been lumped all together as one thing or a couple of species, and they've just been kind of considered an oddity among the large-bodied tamarins, right? So most of the focus has been on the large-bodied tamarins, and then this is just one weird tamarin that happens to be smaller. And so for this study, we use sequences from GenBank, and we use both mitochondrial and nuclear genes. Just to give you a picture, so all of these tamarins that I'm showing you have been classified in the genus Seguinus, and this shows the diversity of large-bodied tamarins, and this is small-bodied tamarins. So you can see many of them have that saddleback, and there are a couple of white ones, but this has been explained as a fairly simple genetic mutation. They may even be considered within the same species as these ones next to them. But creating a time tree to look at the diversity across marmosets and tamarins, we see, right, so this is going, all marmosets and tamarins diversified from each other at about 14 million years ago. And here, this right here, is just the Tamarin group, the Sagwinus group, and you can see that diversification happened at about 9 million years ago. And here we have the small-bodied and the large-bodied tamarins. So they form two distinct separate clades, each with their own evolutionary history. And so again, for the biogeographic analysis, we divided, for this whole marmoset tamarin radiation, we divided up their range into different subregions and tried to see what that history is compared to their current range. And so this is a Biogeobears analysis, and each of the letters at each of the nodes represents whichever subregion here is listed with that letter. And so again, what we found was the Biogeobears analysis suggested that the small and large-bodied tamarins diverged about 9 million years ago from vicariants in this area in the western Amazon. And then if you follow the movement in space across time, you see them separate at first, coming back into sympatry, and both types of tamarin invading the other tamarin's original territory until they have that wide swath of sympatry. And what's interesting when you watch both kinds of tamarins in the same forest together, they'll actually travel together, they forage together, and they may benefit from the different foraging styles that each other has in order to maximize their foraging strategy. Okay. So as a result of this paper, we suggested the splitting of Sagwinus into Leontocebus for the small-bodied tamarins and Sagwinus restricted to large-body tamarins. And this year we formalized this nomenclature with Anthony Rylands leading on that paper. Another thing that was interesting in this paper that parallels what I showed you about the capuchin monkeys is that when you look at the marmoset group, there are Atlantic forest marmosets, Calothrix, and Amazonian marmosets, Miko. And what we found doing the reconstruction here is that the Amazonian and Atlantic forest marmosets diverged at about 5 to 6 million years ago. So this vicariance event that we saw for capuchin monkeys it's mirrored with the vicariance event of Amazon to Atlantic forest with the marmosets as well at the same time period. And then what we also see is if you look here, all of these are Atlantic forest marmosets, but here, this most recent speciation and diversification is moving into the savannah-like habitats with Peninsulata and Calothrix jacus, and that's happening at about 1 million years ago. So just like the capuchin monkeys moved into the savannah-like habitat from the Atlantic forest, at about that same time you also see the marmosets moving into that same type of habitat. So this kind of just reviews what I was saying. I'll move on to another study, which is TD monkeys. The TD monkeys have been considered the most speciose group of neotropical primates, and they're probably also the most understudied. There is very few behavioral studies of them, and there had been really no phylogenetic analysis of them up until this we published this year. And this is Hazel Byrne, is a graduate student at University of Salford, and that was in collaboration with John Bublé from Salford also. And so here you can see the diversity within the TD monkey genus Calocebus. And so what this study aimed to do was try to, this first study that I'm showing you just aimed to do the first phylogeny for TD monkeys to try to understand how these are all related to one another. And so for this study we used 20 nuclear loci and 2 mitochondrial loci, and again we just constructed a time tree and used maximum likelihood and Bayesian analyses. And so what we found here was that there was a long divergence time. So Chiricebus, so what we did in this paper was actually divide Calicebus, which formerly encompassed all TD monkeys, into 3 different genera. Chiricebus, which is the northern Amazon widow TD monkeys, Calicebus, which is now restricted only to the Atlantic forest, and Plectrocebus, which includes all the South Amazon TD monkeys. And you can see that first division between Chiricebus and Calicebus plus Plectrocebus was at 11 million years, and then the second division between Calicebus and Plectrocebus was at 8.3 million years. So if you map this new genus designations onto their distribution, what you can see is that, so what I showed was Chiricebus separated first, diverged first from Plectrocebus plus Calicebus. So it was basically a northern Amazon versus southern Amazon to Atlantic forest split at 11 million years ago. And then the second split between Plectrocebus and Calicebus, so Amazon versus Atlantic forest, is at about 8 million years. So again, we see this this vicariance event at just a little bit earlier between Amazon and Atlantic forest. And so presently I'm working with Hazel Byrne on the biogeography of this. So we're doing a biogeobears analysis to try to understand in more detail these patterns, including this sympatry that you see now and how those species have spread and moved across time. Okay. And then the last example I wanted to give was squirrel monkeys. And this you're going to see a pretty different pattern than the pattern that I showed you for the other 3 groups. And this one was a team effort, again, with graduates, undergraduates, Gohar and Mai here at UCLA, and graduate student Michelle up there from the University of Pará. And then Fernanda, who's pictured here as well. Fernanda Paim is another co-author who works at the Institute of Mamirawa. You can see here they've captured and are taking a blood sample from Cymeri vanzellini, a monkey that was part of this study. So this shows kind of the biodiversity of squirrel monkeys, and squirrel monkeys have been sometimes lumped together as one species, sometimes divided up into as many as about 10 species. And if you look at these drawings, which were done by Amisha Gadani, who's here at the Institute for Society and Genetics, she really spent a lot of time trying to bring out the subtle differences across the species in these drawings. So you can see things like the shape of the crown. Is it straight across, or does it have these arches? Is it gray, or is it black? Do they have bare ears, or do they have tufted ears? How bright is the orange, and how far up the arm does the orange go? These are the kinds of differences that you see that you need to observe to distinguish across these different taxa. Okay? If you were to look at the skeletal morphology, you would have a really hard time distinguishing across any of them. They all look pretty much the same. And so we did a biogeographic analysis for squirrel monkeys, and we found for this group its timeline is much different than the ones I showed you before. All squirrel monkeys started diversifying from a common ancestor at about 1.5 million years ago, and their current range, which is across all the Amazon and a relic population in Central America, all of that spread happened very rapidly in this last 1 million years. And so then just to give you another counterexample, this isn't my own study, but this was also in that special issue of MPE by Di Fiore and colleagues. This is an example of molecular phylogenetic study that instead of splitting taxa apart, lumped taxa together. So originally this was considered Oreonax, and then there are several species of Lagothrix, 2 different kinds of woolly monkeys, but their evidence suggested that they should be subsumed into one genus, Lagothrix. And if you look at the map for these different Lagothrix species, that purple that's in Peru in the Andes region, that's Oreonax, but now Lagothrix flavicauda. And it doesn't have any overlap or sympatry with the other Lagothrix. It's just in a habitat that's distinctly different than where the other Lagothrix are. And so in terms of the spread here, it's about the same timeline as we saw with the squirrel monkeys of just in the last couple of million years they've spread throughout the western Amazon. Does anyone have questions up to now? No? Okay, so basically what I want to do is just to make the argument that we finally have enough data from these different kinds of studies that we can start to construct a comparative biogeography of neotropical primates and try to understand the common patterns and processes that are influencing the biodiversity and the range of these primates. And so I'm just going to pose 2 pretty simple questions as examples of the kind of thing that you can do with comparative biogeography. The first one, how long have modern primates been diversifying in the Amazon and Atlantic forest? And the second one, has there been continuous occupation of savannah-like habitats by neotropical primates? Are they really tropical rainforest specialists that sometimes go out into the savannah, or are they really occupying the savannah in the same way? Okay, and so we want to take a look at this study by Jameson Kiesling that was at the genus level. So they used a genus level phylogeny, they used genomic data, and they divided primates' range basically into these 3 areas, pink being the Amazon area, blue being the savannah-like habitats, and green being the Atlantic forest. And then they did a biogeographic reconstruction at the genus level to try to determine where did the genera diversify, and then where did they move to later. And their overwhelming result, which you can see from all the pink, is that their claim is that genera diversified first in the Amazon, and then only later spread into savanna and Atlantic forest habitats. And so they place Amazon occupation by the extant platyrrhines at 20 million years ago, and say that's been a continuous occupation, and that there's been diversification at the level of genera in the Amazon, and then spreading to the Atlantic forest and to the savanna habitats. And so it does turn out, if you look at all the primates that inhabit the Atlantic coastal forest, then you can find their sister taxon that inhabits the Amazon. So these genus pairs, or pairs of groups that are found in both places. And so what we can do from the kind of studies that I showed you already is map on each of these pairs. When did they diverge? When did the 2 types diverge between the Amazon and the Atlantic forest? And so we get this timeline that basically shows for all of them you have a vicariance event somewhere in the late Miocene to the Pliocene. And so what it looks like is there was continuous occupation of the Amazon. There was a spreading in the mid to late Miocene into the Atlantic forest, and then there was a vicariance event where the Atlantic forest and the Amazon got separated from one another, and then you don't see any movement. The Atlantic forest is just isolated after that. And during that isolation, that's when you see diversification within the Atlantic forest. So these are showing divergence times between North and South Atlantic forest populations. So to answer this question, how long have modern primates been diversifying in the Amazon and Atlantic? It looks like they've been in the Amazon since 20 million years ago, had this later expansion into the Atlantic forest. No new primate genera, at least that are still have living representatives today, entered the Atlantic forest after 4 million years ago. And really the only evidence we have so far from these different studies for animals being able to leave the Atlantic forest and get back to the Amazon is the case I showed you at the beginning of Sapajous, of the robust capuchins. And then the second question, has there been continuous occupation of these savannah-like habitats by neotropical primates? This was one of the figures from the Comparative Biogeography paper, and what it's trying to show is it's kind of trying to go against those assumptions that primates only live in rainforest habitats. So this is bringing out, it's bringing out all the different kind of dry forest and savannah-like habitats that you do find primates in South America. And so then the question is, have they been living there for extended periods of time, or are they always recent immigrants that come in and then get extirpated and come in again? And what's interesting when you do look at this diversity, these are just examples of taxa that can live in these dry habitats. When you look at this diversity, it actually covers across all of these 5 families. So you have squirrel monkeys and capuchin monkeys in dry habitats, night monkeys in dry habitats, marmosets, howler monkeys, and TD monkeys all in dry habitats. So there's multiple instances of neotropical primates figuring out how to live in these other kind of habitat types beyond rainforest. But if you map on the timing, the divergence times between when these sister taxa are found in Atlantic forest or in Amazon, and then you have a move into the dry forest, what you see for all the examples we have information on so far, except for the howler monkey, which seems to be something different going on there. They've been able to colonize dry habitats for about 4 million years at least. All the other species, it's been in the last million years, even some of them even 500,000 years or more recently that they colonized those. And when you look at species distribution or genus distributions, what you often see for other taxa is that there's a huge gap in those dry areas. So they're living in wet forest here, but nothing's in between, suggesting that they've gone in and disappeared. And so the evidence combined together suggests you have this turnover of animals going in, making it sometimes in these dry areas, getting extirpated, but then going back in from the rainforest habitats over time. Okay, and so then the last thing that I wanted to talk about is, what does all this mean? What do these kind of studies mean for taxonomy and for conservation priorities? How does it change things if we learn more information and understand more about biodiversity and evolutionary history? And so it turns out these usual suspects, the same people who are writing the papers and thinking about primate biogeography and primate phylogenetics, they're actually often exactly the same people who are members of the IUCN, who make the decisions about what the conservation status should be for these same neotropical primates. The same invested scientists that care about one part of the story care about the other part of the story too. So this is just a picture from the IUCN meeting to determine conservation status for all neotropical primates. We're at a Thai restaurant in Houston taking a break. And so just to give you an example from the first study I showed you, looking at robust capuchin monkeys. Right now, if you go online, you can see all the different species status on IUCN on this red listing. So we talked about Sapajous xanthosternus in the Atlantic coastal forest. It's considered critically endangered. Another one of those Sapajous species in the Atlantic coastal forest is Sapajous flavius, also critically endangered. But if you take what we said in our paper to be right, Flavius is this one right here that was actually most closely related to these other Amazonian and savannah-like populations. And we suggested that it be encompassed in the widespread Sapojuice apella. And so those particular individuals in those populations that have been called Flavius, if we change the taxonomy to this, this recommendation that we have from our work, then those individuals go immediately from being critically endangered, the highest priority for funding and attention, to least concern. Now they're just a weird variant of this whole huge population. Okay. And so, so I have a graduate student who I'm working with along with Tony Di Fiore at the University of Texas at Austin. She's actually trying to dig deeper down into Flavius versus libidinosus, the savannah-like inhabitant, the capuchin monkey, to see, is this really hybridization? Is there really something different genetically about Flavius? Or is this really just a morphological variant of libidinosus that has gotten isolated and looks blonde because of a few genetic changes that aren't really significant in an evolutionary time scale? Yeah, there's Amelie right there. Similarly with squirrel monkeys, if you take this this number to heart that they've all diversified at 1.4 million years ago, well, it turns out that a lot of geneticists feel that one way to standardize what you categorize as species is to just have a 2 million year cutoff point. And so any diversification that happened after 2 million years, that's just all part of the same species, that's just population variation. So if you went with that kind of genetic framework, then all of these capuchin monkeys, including Vanzolini or Steady, which are both critically endangered now, they're considered that, they would all be lumped into this incredibly cosmopolitan single species of squirrel monkey, and again, least concern. And then on the other hand, these changes like splitting Leontocebus and Saguinus, or splitting Callicebus into 3 different genera, what that tends to do in the conservation community and in the scientific community is bring new interest to this diversity. People suddenly start wanting to know, wow, I never thought about Characebus, I never thought about those widowtides. Let's see what else is going on with them. And so what you see, I saw this also with this split of Sapojuice and Cebus, the gracile and robust capuchins, which up until then people had thought the robust capuchin was just kind of a weird other gracile capuchin that got robust, instead of seeing it as 2 lineages that have diversified separately. And so I've seen dozens of papers come out on Sapojuice after that split, where there really is a renewed interest in understanding that group. So, I think that these changes, whether they're happening at the genus level, species level, subspecies level, these changes matter in terms of research and enthusiasm, and also public funding. And so there's this cycle of the phylogenetic and biogeographic studies affecting the taxonomy, and then the taxonomy directly affecting the conservation status, and then conservation priorities. I wanted to put up this quote. So one of the premier Brazilian taxonomists, primate taxonomists, he goes by Cazuza. His favorite phrase is always more, never fewer. So his idea about studying primate diversity in Brazil is that we always want to find hidden diversity. We always want to illuminate what those differences are. And we never want to lump species back together. We never want to lump genera back together because he doesn't say it's directly because of these conservation implications, but that's the idea, right? You want to always have more, divide more, have more of them critically endangered, so more attention is paid to them and they can get more help. right? And so that kind of brings this, so you saw that there's this play where you're moving back and forth between being the scientist that does the phylogenetic studies and being the conservationist that interprets those studies to decide which species count as what, right? How do you navigate that field? And then finally, I just wanted to put out some other questions that are questions for, not just neotropical primate conservationists, but in general, of this focus on, the more unlikely they are to survive, the more money and focus is placed on them. Is it really worth our money and effort to save all of the biodiversity, or should we be focusing our studies more on those species that have a chance? And so just again, thinking back on the Atlantic forest, the savannah-like habitats, and the Amazon, what you see in Brazil is so much focus has been spent, so much money on reintroductions, on planting, on parks in the Atlantic forest that, as I told you, has been devastated for hundreds of years already. At the same time, while people are focusing on that area, what's happened in the other parts of Brazil? They are rapidly, rapidly declining in habitat availability because of anthropogenic change. So how long do we wait in those areas before things are really in trouble and then finally go in and try to save those last populations? So with that, I would love to discuss some of those ideas, or take questions. Thank you so much for inviting me.
Get the sign-in sheet. Any questions for Jeffrey?
I was just wondering if you could explain in more detail how the beast phylogeography program works and how it reconstructs this ancient.
Okay, so the beast phylogeography, I think, let me just go back to, I would say, so I would say a nice example is this one here, because it's really supposed to be used, it's phylogeography, so it's looking within a species. And so here what we were saying in this analysis, we think that all of this group, that clade 4 that's widespread, is actually one species. And so we use this analysis. What you enter in as information is the locality data for every tip of every individual that you sample. And so these tips are always going to be a single location, so it's going to be a single region that they're coming from. And they're representing those populations in those different regions. And then it's going to take the, that tip data along with the topology, and it includes consideration of the uncertainty in the topology. And you can also enter some information about how close or far different regions are from each other to influence the probability of how likely you can jump from region to region. And then it's reconstructing the ancestral state at each node for where that population came from, or where it was as you go back in time.
Great, thank you.
Mind to follow up? Can I ask that
Yeah,
first?
sure.
I wonder how much then that is biased by the sampling strategies of the researchers. So if the data points which are sort of based on where you're picking up the individuals, and one research group samples in a very small area, and another research group goes out and samples more broadly, how does that affect the way that they're doing?
Yeah, so I mean, so part of in constructing what you want the regions to be are to be making decisions about what you think is important in terms of geographic barriers. So here, the reason that we split the Amazon into 4 segments is because we feel like the Amazon River and the Madeira River and Negro River are the biggest geographic barriers that exist within that frame, right? And so, so you can also do a continuous analysis where you actually put in the coordinates of each sample that you get, or you can lump things together in this discrete analysis where you're just, you have those actual coordinates, but you're putting them into these region blocks. But definitely one concern that we had was this question of bias of where the sampling came from, and that's why we redid the study with 3 times as many samples and locations to see if we would come out with the same result. In this case, we did come out with the same result, but I think the more complete sampling that you can have, the more secure you can be in that the model really seems to be showing what you're expecting it to be a little better.
If and when in the past taxa collapsed into one, have there been like measurable policy or like business effects in terms of whether logging is allowed in an area, for example?
Yeah, I mean, I think it would be an interesting study to do to try to see how taxonomy and conservation status really does impact other kinds of human use of landscape or other things beyond just the conservation status per se. And even there's kind of an assumption or belief in at least the primatological community that if your species is listed higher on the conservation status threat category, that you're more likely to get funding, and that would be something to test as well. Is it really true that those species are the ones that consistently get more funding if they're put higher and higher up on the list, and if they move up and down the list, does the funding change with those moves?
I was wondering whether you could use some of these biogeographic techniques to map onto the behavioral data and try to understand why you might see, say, more specialization or separation of a feeding strategy in some areas than others. So I was interested in what you were saying with the tamarins, where sometimes they go forage and they might benefit from the different feeding strategies than others. So I was wondering with some of the phylogenetic reconstructions that you've done, do you have, say, 2 areas where there's symmetry, and in one case it's been sort of longer than others? Would you expect, so can you map that onto, say, feeding strategy divergence or other kinds of behavioral specialization?
I mean, I think that definitely would be a really interesting application, and as I said kind of at the beginning of the talk, you absolutely can map things like feeding behavior onto a phylogeny to see how that is expected to change over time, and you could do some combination. I mean, I think some interesting things come from these studies in terms of these questions of sympatry. So what it seems like, at least for neotropical primates, and this is not necessarily the same story when you look at something like genins in Africa, where you have very closely related species in sympatry with each other, but at least in the neotropics, it seems in general that there has to be some serious taxonomic shift in behavior or foraging strategy or something. There's enough of a change that they can come back into sympatry with each other and still both be on the same landscape without one or the other population just going extinct. I think an interesting thing that I found with the data that I showed you was that, so remember that the robust capuchin monkeys, at least in the model that I showed, very recently came in and spread around the Amazon. And I showed you that the squirrel monkeys also are a very recent radiation that spread out throughout the Amazon around that same time period. And it turns out that that tufted capuchin monkeys, or robust capuchin monkeys, and squirrel monkeys have the most common associations with each other for any 2 neotropical primate taxa. And the squirrel monkeys benefit from the foraging style. They benefit from cebus in some locations also, but they're more closely aligned with sapajous in terms of benefiting from their destructive foraging style and capturing flying insects that the robust capuchin monkeys scare up. And so this association, I mean, one thing would be interesting to try to model is if this association helped the squirrel monkeys spread along with the robust capuchin monkeys across the Amazon.